Number 1. Haikouichthys. In 1984, a Chinese paleontologist named Hu Xiangguang was leading a field expedition in Yunnan province. His students had stopped for lunch. Hu wandered off alone into the hillside above the Qingjiang fossil beds. And in the exposed mudstone, he found something that would eventually rewrite the origin story of every animal with a backbone. The Qingjiang biota, as the site came to be known, has since produced over 200 species. Many researchers consider it more significant than the Burgess Shale. But the fossil that matters most for this list was not described until 1999, when Shu Deegan and Simon Conway Morris published their analysis in Nature. They called it Haikouichthys. It was 25 millimeters long, roughly the size of a paperclip, 530 million years old, early Cambrian, and it had a notochord, a primitive spinal cord. That single feature made it one of the earliest known chordates, the phylum that includes every fish, every amphibian, every reptile, every bird, and every mammal alive today, including you. Haikouichthys had no jaws, no paired fins, no mineralized skeleton, but it had V-shaped muscle blocks called myomeres. The same basic structure found in the body wall of every fish swimming in the ocean right now. It had paired sensory structures that researchers interpret as primitive eyes. It had pharyngeal gill slits. The preservation was extraordinary. Fine Cambrian mudstone captured soft tissue impressions well enough to identify internal organs in an animal smaller than your thumb. Before this discovery, the oldest accepted vertebrates were Ordovician jawless fish, conodonts and arandaspids, dated to roughly 480 million years ago. Haikouichthys pushed the origin of all vertebrates back by at least 30 to 50 million years. That is not a minor adjustment. That is a fundamental rewrite of when the backbone first appeared on Earth. There is a competing candidate, Milocunmingia, found in the same Xinjiang beds described by the same team. The debate about whether it is actually the same genus as Haikouichthys continues. Jun Yuanqin at the Nanjing Institute of Geology and Paleontology provided additional specimens in the early 2000s, but the core conclusion has held. The mainstream interpretation is clear. Haikouichthys is a basal vertebrate. It confirms that vertebrates diversified rapidly during the Cambrian explosion alongside arthropods, mollusks, and every other major animal phylum. The entire backbone of animal life appeared in a window of perhaps 20 to 25 million years. Not everyone agrees on the mechanism. Some researchers argue the Cambrian explosion is partly an artifact of preservation bias. Vertebrates may have originated earlier in the Ediacaran period, but their soft bodies left no trace. Andrew Parker's light switch hypothesis takes a different angle entirely. He proposed that the sudden evolution of eyes triggered an arms race that drove the explosion. Haikouichthys, with its primitive visual structures, could be evidence for that trigger. Modern molecular clock estimates broadly agree with the fossil. Most place vertebrate origins in the early to mid-Cambrian. A few push it further back. But the physical evidence starts here. In a paperclip-sized fish in a Chinese hillside, this is the root of the vertebrate tree. Everything with a spine traces back to something like this, but where the tree's youngest branch, the human branch, actually began, turned out to be completely wrong. Number 2, Sahelanthropus tchadensis. On July 19, 2001, a Chadian University student named Ahunti Jimdumalbaye was prospecting in the Jurab Desert of northern Chad. He was part of the Messian Paléoanthropologique Franco-Chadienne, a joint French-Chadian research team. And in the sand and rock of one of the most inhospitable places on Earth, he found a skull. It was over 2,500 kilometers west of the East African Rift Valley. That matters, because at the time, virtually every paleoanthropologist on the planet believed human evolution had occurred in East Africa, specifically along the rift. The dominant framework was called the East Side Story. Proposed by Yves Copins in 1994, it argued that the formation of the Great Rift Valley created a geographic barrier. Apes evolved in the wet forests to the west. Hominins evolved on the dry savannas to the east. Clean, elegant, and as of July 19, 2001, wrong. The skull was nicknamed Toumaï. It means hope of life in the local Goran language, a name traditionally given to children born just before the dry season. Michel Brunet and his colleagues described it in Nature in 2002. Specimen number TA 266-01-060-1, dated to between 6 and 7 million years ago. Potentially the oldest known hominin. The morphology is a mosaic, a flat face, small canines, a foramen magnum, the hole where the spinal cord exits the skull, positioned underneath, suggesting an upright posture. But the brain case is chimpanzee-sized. Roughly 360 cubic centimeters. This combination of features is exactly what you would expect near the divergence point between the human and chimpanzee lineages. Molecular clocks estimate that split at 6 to 8 million years ago, but the controversy was immediate. Milford Wolpoff and colleagues argued in 2002 that Sahelanthropus might not be a hominin at all. It could be an ancestral gorilla. The cranial features, they said, were ambiguous enough to support either interpretation. Then there was the femur, a thigh bone. Specimen TM266-01-063 was found at the same site. It was kept largely unpublished for nearly 20 years. When researchers finally studied it, a bitter dispute erupted. Roberto Macchiarelli argued the femur indicated quadrupedal locomotion. Frank Guy and Guillaume Daver published a study in Nature in 2022 arguing it supports habitual bipedalism. The debate is not settled. Brunet's team had worked in Chad for over 20 years, temperatures exceeding 50 degrees Celsius. The Jurab was not a desert 7 million years ago, it was a lakeside environment. The associated fossils include fish, crocodiles, and freshwater mollusks. Toros Manala, the discovery site, has yielded over 1,500 vertebrate fossils from the same period. Competing candidates exist. Auroran tuganensis, found in Kenya in 2000 by Brigitte Senut and Martin Pickford, is dated to approximately 6 million years ago. Ardipithecus kadaba from Ethiopia dates to 5.2 to 5.8 million years ago, but Toumaï remains the strongest challenger for the title of oldest hominin, and its location, thousands of kilometers from the rift, suggests some researchers were never looking in the wrong era. They were looking in the wrong place entirely. A speculative but increasingly discussed idea takes this further. Perhaps the human-chimp split was not a localized event at all. Perhaps it was geographically widespread across equatorial Africa. Toumaï would not be anomalous in this model. It would be central. Toumaï proved the map was wrong. The next fossil proved that if you trust the map, the theoretical map, you can find exactly what you are looking for. Number 3, Tiktaalik. Neil Shubin had a prediction. He needed a specific kind of rock, late Devonian age, freshwater deposits, exposed at the surface where a team could actually reach them. The evolutionary models said a transitional form between fish and land-dwelling tetrapods should exist somewhere in sediments from roughly 375 million years ago. Shubin just had to find the right rocks. He found them on Ellesmere Island, Nunavut, Canadian Arctic, approximately 600 miles from the North Pole. His team began searching in 1999. They searched for 5 full field seasons. They found nothing of significance for 4 of them. Then, in 2004, they found Tiktaalik. Described by Shubin, Edward Deichler, and Ferris Jenkins Jr. in Nature in 2006. The name means large freshwater fish in Inuktitut, chosen in consultation with Nunavut elders, and it was a large, approximately 2.7 meters, 9 feet, much bigger than most popular depictions suggest. The anatomy is extraordinary. A flat crocodile-like skull with the eyes positioned on top, adapted for looking upward out of shallow water. The first known fish with a mobile neck, its head could turn independently of its body, no fish before it could do that. And in its pectoral fins, wrist-like bones. Bones that allowed it to prop itself up and do push-ups on a muddy riverbed. A transitional structure, half fin, half limb. It kept its fish features, scales, fin rays, gills. But it had tetrapod features too. A flat head, ribs structured to support the body out of water, lungs inferred from the ribcage architecture. And in 2014, a study published in PNAS by Shubin's team revealed something nobody had seen before. Tiktaalik had a well-developed pelvis and hind fins. This pushed the evolution of hip-driven locomotion back into fish. Before tetrapods even existed, Ellesmere Island sits in the high Arctic now. But during the late Devonian, it was near the equator. roughly 10 degrees north latitude, a warm subtropical floodplain crisscrossed by shallow rivers and streams. That is the environment Tiktaalik inhabited. And it is exactly the environment the models predicted. Tiktaalik filled a precise gap between Haikouichthys, a more fish-like form, and Archaeopteryx, a more tetrapod-like form. The sequence from water to land was no longer a gap. It was a gradient. Richard Cloutier's team complicated the picture slightly in 2020 when they described Elpistostager from Miigwech, Canada, with even more complete finger-like bones. Per Alberg and others have suggested the transition may have involved multiple lineages independently experimenting with terrestriality, not a single ladder, more of a bush. Farish Jenkins Jr., the Harvard professor who co-led the expedition, died in 2012. He never saw many of the later analyses published. Shubin wrote the popular book Your Inner Fish in 2008, later adapted into a PBS documentary. The book's core argument is built on Tiktaalik. It is one of the most compelling examples in all of paleontology of a transitional fossil that was predicted before it was found. Not stumbled upon. Not dug up by accident. Predicted, located, extracted. Tiktaalik proved how vertebrates conquered land. But once they were on land, they started doing something nobody anticipated. They started experimenting with what their bodies looked like. And the next fossil proved that everything we thought we knew about that appearance was wrong. Number 4. Sinosauropteryx. In 1996, a farmer named Li Yumin was digging in Yixian Formation in Liaoning Province, northeastern China. He found a small fossil, a slender, long-tailed creature about 1 meter in length. When paleontologists Ji Chang and Ji Shuan described it later that year, the announcement sent shockwaves through the field. This was the first dinosaur ever found with direct, unambiguous evidence of feather-like structures preserved on its body. Sinosauropteryx prima, a small compseuchthid theropod, dated to between 124 and 130 million years ago. Early Cretaceous. The filamentous structures covering its body were immediately controversial. Some researchers argued they could be degraded collagen fibers rather than protofeathers, preservation artifacts, not biological integument. Philip Currie was among the first Western paleontologists to examine the specimen directly. He confirmed the structures were genuine, but it was not until 2010 that the fossil truly changed what we know. Fuqing Zhang, Mike Benton, and colleagues published a study in Nature analyzing the melanosomes preserved inside the filaments. Melanosomes are microscopic structures that contain pigment. Different shapes produce different colors. The team found 2 types, elongated eumelanosomes and spherical phaeomelanosomes. The conclusion: Sinosauropteryx had a reddish-brown and white striped tail. It was the first dinosaur in history whose color was determined from direct physical evidence. Not speculation, not artistic license. Evidence. Then in 2017, Fianne Smithwick and colleagues published a study in Current Biology revealing something else. Sinosauropteryx had counter shading, darker pigmentation on top, lighter underneath. This is a camouflage pattern used by modern prey animals living in open habitats. Deer have it, rabbits have it. It implies predation pressure. The ecological implications were immediate. Sinosauropteryx was not living in dense forest. It was in open environments, being hunted. One specimen even preserves the remains of a small mammal and a lizard in its stomach. Predator and prey captured in the same fossil. The Yixian Formation is a Lagerstätte, a site of exceptional preservation. Animals were buried rapidly in fine volcanic ash, capturing soft tissue in extraordinary detail. Before Sinosauropteryx, the connection between dinosaurs and birds was based primarily on skeletal similarities. Thomas Henry Huxley had noted these resemblances in the 1860s. But skeletal similarity is not the same as direct evidence. The resistance was fierce. Larry Martin and Alan Fiducia led the band camp. Birds are not dinosaurs. They argued the filaments were not feathers, that birds descended from a different archosaur lineage entirely. But the Liaoning fossils kept coming. Microraptor, Yutyrannus, Genuine Long, Anchiornis, over 40 species of feathered dinosaurs from the Yixian Formation alone. By the mid-2000s, the debate was effectively over. One of the more provocative ideas to emerge from this wave of discoveries is that feathers evolved first for insulation or display, not flight. Feathers may have been repurposed for aerodynamics millions of years after they first appeared. If true, then flight was not the reason feathers exist. Flight was an afterthought. Sinosauropteryx changed what dinosaurs look like. Permanently. But the next fossil changed what our own ancestors looked like, and a tiny bone in a child's throat opened a question that nobody was expecting. Number 5. Dikika Baby. On December 10, 2000, Zeresene Aldamseged was surveying a hillside in the Dikika area of the Afar region, Ethiopia. He spotted the face of a small skull eroding out of sandstone. It took him 5 years to extract it, working with dental tools, grain by grain. What emerged was the most complete skeleton of any early hominin child ever found, specimen DIK1-1-1, Australopithecus afarensis, the same species as Lucy, but this individual was not an adult. She was approximately 3 years old at death based on dental development, and she was 120,000 years older than Lucy herself, dated to 3.3 million years ago. She was nicknamed Selim, peace in Amharic. The media called her Lucy's baby, though she predates Lucy by over 100,000 years. The skeleton is remarkable for its completeness. A nearly intact skull, both scapulae, both clavicles, much of the vertebral column, ribs, fingers, a kneecap, a foot. For a fossil this old, this level of preservation is almost unheard of. Lucy herself is only about 40% complete, but the most startling element is a tiny horseshoe-shaped bone found in the throat region, a hyoid. This bone anchors the tongue and the muscles involved in swallowing and vocalization. In modern humans, the hyoid is essential for speech. It is almost never preserved in the fossil record because it is small, fragile, and not attached to any other bone. Selim's hyoid more closely resembles that of African apes than modern humans. That does not settle the question. It opens it. If a 3.3 million-year-old Australopithecus had a hyoid, what could it do with it? Nobody is arguing for language. But complex vocalizations, calls more sophisticated than a chimpanzee's? The mainstream interpretation is cautious. The hyoid is ape-like. But its very presence invites speculation about proto-vocal capabilities far earlier than previously considered. The rest of the skeleton tells a story of an animal living between two worlds. The scapulae resemble those of a young gorilla more than a human child. The fingers are curved. Both features suggest significant time spent in trees, but the lower limbs are adapted for bipedal walking. A 2018 study in Science Advances examined the foot. It had a bipedal arch, but also a grasping big toe. This child could walk upright on the ground and grip branches with her feet. CT scanning at the European Synchrotron Radiation Facility in Grenoble revealed developing adult teeth still inside the jaw. The brain was approximately 1/3 the size of a modern human's. The mix of arboreal and terrestrial adaptations has led to ongoing debate. Some researchers argue the climbing features are functional, others say they are evolutionary baggage, vestigial holdovers from a more arboreal ancestor. The truth likely lies somewhere in between. Selim changed how scientists understood childhood in early hominins. The prolonged period of dependency implied by her anatomy, an animal that both climbs and walks, that needs care in both worlds, mirrors something we see in modern human children. Dependence, vulnerability, a long time spent learning. Selim was caught between the trees and the ground. The next fossil was caught between two completely different ways of conquering the sky, and it chose the one that failed. Number 6. Yiqi. In 2007, a farmer in Hebei province, China, found a small fossil in the Tiaojishan Formation. It sat in a collection for years before Xu Xing and Jing Xiaoting described it in Nature in 2015. When they did, it defied categorization. Yiqi. Pronounced Yiqi, it means strange wing in Mandarin. It is the shortest valid dinosaur name in scientific nomenclature. The animal was about the size of a pigeon, approximately 380 grams, a Scansauropteryx theropod, dated to 160 million years ago, late Jurassic, roughly 10 million years before Archaeopteryx. It had feathers on its body, That was expected by 2015. What was not expected was the elongated rod-like bone extending from each wrist, a steliform element, unique among all known dinosaurs. And alongside those bones, patches of preserved membrane tissue. Yichi did not fly with feathered wings. It flew with membranous wings, like a bat. This was not supposed to happen. The entire narrative of flight evolution in dinosaurs centered on feathers. Feathered wings led to birds. That was the story. Yichi proved there was a second story running in parallel. Dinosaurs independently evolved at least two completely different flight mechanisms: feathered wings in the Monoraptoran lineage, and membranous wings in the Sinosauropteryx. A 2020 biomechanical study by T. Alexander Desecchi and colleagues in iScience tested how well Yichi could actually fly. The answer was not well. It was a glider at best, and a poor one. Worse than a modern flying squirrel. The membrane wing design did not generate enough lift for powered flight. It was by any measure an evolutionary dead end. This was confirmed in 2019 when Wang Min and colleagues described Ambopteryx longibrachium, another Sinosauropteryx, also with membranous wings. This was not an anomaly, it was a family trait. The entire lineage was running the membrane experiment, and the entire lineage went extinct by the end of the Jurassic. The experiment lasted perhaps 10 to 15 million years. Meanwhile, feathered wings went on to produce every one of the more than 10,000 bird species alive today. Some researchers have questioned whether the styloform bone was actually part of the living animal. The preservation is ambiguous enough that taphonomic artifact cannot be fully ruled out. But the discovery of Ambopteryx with the same structure makes this objection increasingly difficult to maintain. Shuxing, who described Yichi, has named over 70 new dinosaur species over his career. He is one of the most prolific taxonomists in the history of paleontology. The Tiaozhishan Formation, where Yichi was found, has become a crucial window into middle to late Jurassic ecosystems. And Yichi has become a powerful illustration of a principle that runs through all of evolutionary biology. Evolution does not find one solution and optimize it. It runs multiple experiments simultaneously. Most of them fail. The failures are not footnotes. They are the story. Yichi tried membranes and lost. But the question of how feathered flight actually worked, whether the first feathered flyers could truly fly at all, remained unresolved for 160 years. It took a particle accelerator to settle it. And the fossil it was pointed at is the most famous one in the world. Number 7. The Dmanisi Skulls. In 1991, a Georgian archaeologist named David Lord-Kipanidze was excavating the medieval town of Dmanisi in the Kvimo-Kartli region of Georgia. He was looking for artifacts from the Middle Ages. What he found was a hominin jawbone nearly 2 million years old. Between 1991 and 2005, his team recovered 5 skulls designated D2280, D2282, D2700, D3444, and D4500, known collectively as Skull 5. They were dated to between 1.77 and 1.85 million years ago, making them the oldest known hominin fossils outside of Africa. The skulls are wildly variable. Brain sizes range from 546 cubic centimeters in D2700 to 730 in D4500. Skull 5, described by Lord-Kipanidze and colleagues in Science in 2013, has a massive protruding jaw, a tiny brain, and a face unlike any other early Homo specimen. It is the most complete early Homo skull ever found, and it is profoundly strange. Here's the problem. If these 5 skulls had been found at 5 different sites on 5 different continents, they would almost certainly have been classified as 5 different species: Homo habilis, Homo rudolfensis, Homo ergaster, Homo georgicus. The variation between them is that large, but they were not found on different continents. They were found in one place, in one population, at one time. The 2013 Science paper made the argument explicit. If this much morphological variation can exist within a single local population, then many of the named species in the early Homo lineage may not be real species at all. They may be normal variation within one highly variable lineage. The implication is that the human family tree needs to be drastically pruned. Perhaps habilis, rudolfensis, ergaster, and georgicus should all collapse into a single wide-ranging species of early Homo erectus. Not everyone agrees. Ian Tattersall, Jeffrey Schwartz, and Fred Spoor have pushed back. They argue Dmanisi could represent a uniquely variable local population, a founder group, an outlier. The morphological differences between established species from African sites, they say, are real and cannot be explained away by one Georgian sample. But Dmanisi offers more than a taxonomic puzzle. One individual, D3444, was an elderly adult who had lost all but one tooth long before death. The bone had resorbed. This person could not chew. Someone fed them. Someone kept them alive. This is one of the earliest known examples of care for the infirm in the hominin record. The site sits at 1,171 meters elevation at the confluence of two rivers. It was a crossroads. The associated fauna includes saber-toothed cats, giant ostriches, and short-necked giraffes. The stone tools are Oldowan. Mode 1. The simplest technology known. Dmanisi is the strongest evidence that early Homo left Africa far earlier with far smaller brains and far simpler tools than previously assumed. The old model was that migration required big brains and advanced technology. Dmanisi says otherwise. Small-brained hominins with the most basic stone tools made it to the edge of Europe nearly 2 million years ago. Some researchers speculate this was a founder population, a small, highly variable group that pioneered the out-of-Africa route. If true, the variation is not anomalous. It is exactly what you would expect. Dmanisi showed that early humans were more variable and more mobile than the models predicted. But the next discovery challenged something even more fundamental. The assumption that nothing biological can survive 68 million years inside a bone. Number 8. Schweitzer's T-Rex soft tissue. In 2005, Mary Higby Schweitzer was working on a Tyrannosaurus rex specimen at the Museum of the Rockies in Montana. The specimen, MOR 1125, had been excavated from the Hell Creek Formation by Jack Horner's team. It had to be broken to fit into a helicopter for transport. Schweitzer took fragments of the shattered femur back to her lab. She dissolved the mineralized bone in a weak acid solution, and inside, she found something that the entire field of paleontology said could not exist. Flexible, transparent blood vessels, branching structures that looked like circulatory tissue, red-brown bodies resembling red blood cells, fibrous material with the texture of soft tissue. The specimen was 68 million years old. The results were published in Science in March 2005. The reaction was immediate and overwhelmingly skeptical. The prevailing dogma was absolute. All original organic material in a fossil is completely replaced by minerals within approximately 1 million years. That was not a guideline. It was treated as a law. What Schweitzer was claiming violated it by a factor of 68. She had actually seen something similar before. In 1993, while studying a different T-Rex leg bone, she had noticed round red structures inside blood vessel channels. She mentioned it to Jack Horner. She was told to prove they were not red blood cells. The field moved on. She did not. In 2007, Schweitzer published protein sequencing results in Science. Her team had extracted collagen fragments from the T-Rex bone. The proteins were most similar to chicken collagen. This was the first molecular evidence drawn from actual dinosaur tissue that directly supported the evolutionary relationship between dinosaurs and birds. The pushback came fast. In 2008, Thomas Kay and colleagues published a rebuttal in PCLOS1. They argued the structures Schweitzer found were not original tissue, they were bacterial biofilm. Colonies of microbes that had colonized the bone after burial and formed structures that mimicked blood vessels. Schweitzer's response was methodical. Her team ran immunological tests. They used antibodies specifically designed to bind to collagen, hemoglobin, and other proteins. The antibodies bound to the dinosaur tissue. If the structures were bacterial, the antibodies should not have reacted. In 2009, she found similar soft tissue in an 80 million year old Brachylophosaurus canadensis specimen. It was not a one-off, it was not contamination, it was a pattern. By 2013, Schweitzer had proposed a mechanism. Iron from degrading hemoglobin generates free radicals. Those free radicals cross-link proteins, effectively tanning them like leather. Her lab experiments demonstrated this directly. Ostrich blood vessel tissue soaked in an iron-rich hemoglobin solution remained intact for over 2 years at room temperature. Untreated tissue decayed within days. The finding has been seized upon by young Earth creationists as evidence that fossils are not millions of years old. Schweitzer, a devout Christian, has publicly and repeatedly rejected this interpretation. Her faith, she has said, does not conflict with mainstream geology. As of the mid-2020s, similar soft tissue findings have been reported from multiple specimens across different geological periods and locations. The question is no longer whether it is real. The question is how common it is, and what else might be preserved that nobody has thought to look for. Some researchers speculate that recoverable DNA fragments could exist under the right conditions. That remains highly debated. But the door Schweitzer opened is not closing. She faced years of professional resistance. Colleagues told her the findings were impossible. Therefore wrong, she proved them wrong with chemistry. She opened an entirely new field, deep-time molecular paleontology. And the irony was not lost on anyone that her PhD advisor, Jack Horner, was the scientific consultant for the Jurassic Park films. Schweitzer found 68-million-year-old molecules inside a bone. The next fossil presented a different kind of impossibility. A wolf with hooves. That turned out to be the ancestor of every whale in the ocean. Number 9. Pakicetus. In 1981, Philip Gingerich and Donald Russell described a new genus from fragmentary skull material found in the Kuldana Formation of Punjab, Pakistan. They called it Pakicetus, dated to approximately 50 million years ago. Early Eocene. Based on the skull fragments and teeth, Gingerich initially reconstructed it as an amphibious, seal-like creature, something that split its time between land and water. He was half right. In 2001, G. M. Thewison and colleagues published a much more complete skeleton in Nature. The picture changed completely. Pakicetus was not seal-like. It was wolf-like, fully terrestrial. 4 long legs, hoofed feet, approximately the size of a large dog. There was nothing about its postcranial anatomy that suggested an aquatic lifestyle. Nothing you could see from the outside. The evidence was in its ears, specifically the tympanic bulla, a bone in the middle ear. In all living whales, this bone is involuted, rolled inward, and unusually dense. This configuration is diagnostic. No other group of mammals has it. Pakicetus had it, a wolf-shaped animal running around on hooves in what is now Pakistan, with whale ears. Additional evidence came from the teeth. The arrangement of Pakicetus's triangular molars matches the pattern seen in later, progressively more aquatic whale ancestors, the Archaeopteryx. And molecular evidence, accumulating through the 1990s and 2000s, confirmed the broader relationship. DNA studies showed that whales are nested within the order Artiodactyla. Their closest living relatives are not seals, not sea lions, hippos, pigs, deer. Whales are even-toed ungulates that went back to the ocean. The fossil record of this transition is one of the most complete in all of evolutionary biology. Pakicetus, terrestrial, approximately 50 million years ago. Ambulocetus natans, described by Thewison in 1994, a 3-meter crocodile-like creature that could both walk and swim, at 49 million years ago. Couchicetus, otter-like, at 46 million years ago. Rhodhocetus, seal-like, also 46 million. Basilosaurus, fully aquatic, serpentine, at 40 million years ago. Then modern cetaceans. 10 million years. Every major step preserved. The Kuldana Formation represents the shallow shoreline of the ancient Tethys Sea, which covered much of what is now Pakistan and India. This warm, shallow coastline was the crucible of whale evolution. Nearly all of the key transitional whale fossils have been found in Pakistan and India. The Tethys gave us whales. What drove a terrestrial predator into the water? One hypothesis, advanced by Thewison and others, is competition. Early carnivorans were diversifying rapidly on land during the Eocene. Pressure from these competitors may have pushed certain artiodactyl lineages toward aquatic niches. Pakicetus likely hunted near water. Something like a modern tapir or fishing cat. It was already at the water's edge. It just had not gone in yet. Gingerich initially faced skepticism. The idea that a hoofed wolf-like animal was ancestral to whales seemed absurd. The earbone evidence proved decisive before the full skeleton sealed it. Hans Thewissen later wrote the definitive popular account of this transition in his 2014 book, The Walking Whales. A wolf became a whale in 10 million years, and the evidence is all there. Every step, every intermediate form. It is one of the most powerful answers in all of paleontology to the claim that there are no transitional fossils. But the last fossil on this list is a different kind of lesson. It is a reminder that even 160 years of study is not enough to fully understand a single specimen. Number 10, Archaeopteryx. In 1861, quarry workers in the Solnhofen limestone of Bavaria, Germany, found a fossil that would become the most famous in the history of paleontology. Archaeopteryx lithographica, a creature with the feathered wings of a bird and the teeth, bony tail, and clawed fingers of a reptile. 150 million years old, near late Jurassic. It was described the same year. Within 2 years, it was at the center of a global debate about the relationship between dinosaurs and birds. That debate has never fully ended. 13 specimens have been recognized as of the mid-2020s, discovered over a span of more than 160 years. Each one has added something, each one has shifted the interpretation. The 12th specimen, found in 2011 and described by Oliver Rauhut and colleagues in 2018, came from the Painton Quarry, a slightly different and younger geological horizon than the classic Solnhofen sites. But the truly transformative work was not about finding new specimens. It was about applying new technology to existing ones. In 2019, Dennis Votin and colleagues used synchrotron microtomography at the European Synchrotron Radiation Facility in Grenoble, France. They scanned the flight feathers of multiple Archaeopteryx specimens. A synchrotron is a type of particle accelerator. It produces beams of X-rays millions of times brighter than a hospital machine. And for the first time, it revealed the internal microstructure of the primary flight feathers. The geometry of the rachises, the shafts, they were thin, significantly thinner and weaker than those of any modern bird capable of sustained flapping flight. The downstroke that an albatross or an eagle uses to power through the air, Archaeopteryx could not do it. The feather structure would not support it, but the feathers were asymmetric, one vein wider than the other. This is an aerodynamic feature. It generates lift. Feathers built purely for display or insulation are symmetrical. Archaeopteryx's were not. It was doing something with those wings, just not sustained powered flight. The conclusion was nuanced. Archaeopteryx was an intermittent flyer, capable of short powered bursts, gliding, possibly wing-assisted incline running, a behavior observed in modern ground birds like chukar partridges. A 2018 study by Vatin, published in Nature Communications, examined the wing bone cross-sections. They showed compact cortical bone, similar to modern burst flyers like pheasants and quail, not to sustained flyers like albatrosses or hawks. This overturned both competing camps. One side had argued Archaeopteryx could fly well. The other said it could not fly at all. The answer was more subtle than either position allowed. The fossil has survived even stranger controversies. In the 1980s, Fred Hoyle and Chandra Wickramasinghe publicly claimed the London specimen was a forgery. The feather impressions, they argued, had been artificially pressed into the stone around a genuine reptile skeleton. Multiple independent analyses, ultraviolet imaging, synchrotron scanning, chemical mapping have debunked this conclusively. In 2019, Ryan Carney re-examined the very first Archaeopteryx specimen, the original 1861 feather. Using fluorescence imaging, he determined it was black, not white, not gray. Black. Even the color had been wrong. Archaeopteryx has been classified as a bird, reclassified as a non-avian dinosaur, then reclassified again. The latest consensus places it at or very near the base of Avialae, the bird lineage. But whether it is a true bird or a very close outgroup remains debated. After 160 years, the most studied fossil in history still cannot be definitively placed. That is not a failure of science. It is an illustration of how science works when the evidence is genuinely complex. Each new technology reveals something new. Each new specimen shifts the balance. The Solnhofen limestone formed in a tropical lagoon, small islands surrounded by warm, shallow seas. Archaeopteryx lived there 150 million years ago, and it is still teaching us things. 10 fossils. 10 assumptions overturned. A paperclip-sized fish proved vertebrates were already part of the Cambrian explosion. A skull in the wrong desert proved human origins were not confined to East Africa. A fish with wrists proved the land invasion could be predicted. A farmer's fossil proved dinosaurs wore feathers and that their color could be measured. A 3-year-old skeleton proved our ancestors were caught between the trees and the ground. A pigeon-sized dinosaur proved there was more than one way to fly, and that most ways fail. 5 skulls in Georgia proved the human family tree may have too many branches. A cracked bone proved organic molecules can survive 68 million years. A wolf proved it was the ancestor of every whale in the ocean. And the most famous fossil in history proved that 160 years of study is not enough. The pattern is consistent. The fossils that changed everything were not the ones that confirmed what scientists already believed. They were the ones that contradicted it, that broke the models, that forced the textbooks to be rewritten. Each one met resistance, skepticism, accusations of contamination, claims of forgery, dismissal. In every case, the evidence prevailed, slowly, painfully, but completely. The fossil record is still overwhelmingly incomplete. By most estimates, we have found fossils of perhaps 1% of the species that have ever lived, maybe less. That means the next fossil that changes everything is almost certainly already in the ground. It might be found by a paleontologist who trusted a theoretical model, like Neil Shubin walking the Arctic for 5 years, or it might be found by a farmer who had no idea what he was looking at, like Li Yuman turning over a rock in Liaoning. The story of life on Earth is not finished being told. We are still one fossil away from having to rewrite everything again. And that is not a weakness of science. That is the entire point.